Hi everyone, my name is Marta Anguiano. I'm a senior in the Chemical and Environmental Engineering department, and today I'll be presenting on the characterization of ultrafine bead patterns and distributions in mouse lungs. So to start off, I want to go over why the lungs are important. So we have different components in our respiratory system. We have the trachea here, our bronchi, and the bronchioles, which take us into the two different lungs. And deeper in the lungs, we have our alveoli. This is a small little diagram of what the alveolar sacs would look like, and we have millions in our respiratory system. And so the most vital function of the respiratory system occurs in this site, which is gas exchange. And so when we have CO2 in our body, it essentially gets exchanged for oxygen that we're breathing in. And this is where it occurs. CO2 leaves and oxygen gets diffused into our lung capillaries. And that's how the blood cells are able to carry oxygen throughout our bodies. And so, as I briefly mentioned, with these very different components and subcomponents of the respiratory system, they do have varying areas. The trachea is going to be much larger than the bronchi and the bronchioles. So as a result, these structural changes are going to require certain support systems. Specifically looking at the airways and the alveoli, the airways do have a ciliated epithelium lining, and this does help with it does help with removing mucus or foreign objects in the airways. This is primarily due to the cilia that have the flexibility and ability to move in a rhythmic motion. And so unlike the airway epithelium, the alveoli have macrophages, which are white blood cells. And these primarily serve as the first line of defense against invading pathogens. And this is an image of what the cilia hair-like structure looks like. And then this is what the macrophages look like. And so by keeping this in mind on the different support systems that the airways have and then the alveoli have, we can go into the different health effects that come from particulate matter exposure. So particulate matter are characterized based off of either their chemical properties or the aerodynamic diameters. So for my study specifically, we're going to be focusing on the diameters. And so this can be, for coarse particles specifically, they have a diameter range of 2.5 to 10 microns in range. For fine particles, which is where my study falls under, the range is between 0.1 to 2.5 microns. And then for ultrafine particles, this is anything less than 0.1 microns. And so they can generally be referred to as either PM10, PM2.5, and ultrafine particles. PM2.5 and ultrafine particles do have the ability of reaching the alveolar region due to their small diameter size, and the associated health effects range from having a cough, reduced expiratory flow, and having an increase in hospital admissions for those of us who have asthma or for just any medicine usage. So for the larger particles, this deposition in the respiratory system is going to remain in the upper respiratory regions, and they do still have associated health effects. Usually they are associated with respiratory diseases, inflammation, and cardiovascular diseases. However, studies have shown that the impacts of these particles are not as impactful as ultrafine or finer particles are. So intranasal administration is a widely used technique used across various fields, specifically with what we have discussed so far. It's a popular technique because of its non-invasive, because it's not invasive, sorry. So this can either be used to test medicine efficiency to treat various diseases and inflammation in the respiratory system, or it can also be used to induce respiratory inflammation to observe this inflammation or the inflammatory cell responses in different models. However, it does have a major implication, which is that it is not reflective of a natural route of exposure. This will lead to huge implications for intranasally administrated studies, where the immune responses in animal models such as mice are not going to be representative of what we actually observe in actual exposure studies with human beings. So therefore, in our lab, we do have an aerosol chamber, aerosol chamber that's gonna take away this implication, as it does provide a more natural route of exposure. And so in our study, we do have two main questions. The first one is, can the bead deposition patterns be detected through a histological method? The second is, can an analytical method be developed to characterize these different patterns observed? So for the first question, I do want to mention that in this study, we do use fluorescent beads. The reason being is because they do fluoresce red, and as we can see in these images, you can clearly see where the beads are depositing. You can see these depositing here, and then at a further magnification at 30x, you can even see these beads here in this airway and in the alveoli. And this is primarily why we use fluorescent beads to expose the mice. And so along with that, to answer our other questions, for our methods, we expose the mice to these beads using either the aerosol chamber, where they will continuously inhale these beads, or intranasally, as it is widely used in various studies. We will then go on and dissect the mice to extract their lung lobes, fix them, and slice them. Then images are taken using the Keon's microscope. And to answer question 2, we actually incorporated the Python grid analysis method, which I will be covering later on in the presentation. So from these various studies, as I mentioned, we do have intranasal exposures and aerosol exposures. So for intranasal exposures, we do have a small dose exposure where the mice were administered to a total of 20 microliters. And on the left side of the slide, you can see that we have a large airway and the smaller airways here. But in these images, you mostly just see the tissue fluorescing. We don't really see a lot of bead deposition here. Whereas in the alveoli images, although it's very small to see here, we do have about 1 or 2, maybe 3 beads depositing in the alveoli. So overall, for the small dose intranasal exposure, we didn't really see much deposition, but we did see a few beads here and there. And so as a result, we decided to create the large dose intranasal exposure. So for this exposure, the mice were administered to a total of 40 microliters, which did change the patterns observed. For this overview image of the mouse lung, in the upper region, we do see many clusters and a lot of different beads deposited. And these images here show the alveoli and the airways at 30x magnification. And so again, you can see these very big clusters here being deposited into the alveoli regions. This here is going to be an airway, and we do see a big cluster here of beads. And we do have another airway here showing that the beads did deposit here as well. And this was the main pattern observed for the large dose exposure. For this slide, I mostly want to show that for other regions of this same lung lobe, we did not see any deposition. These are alveolar regions, and this is an airway here from another part of the lung lobe. And as you can see, we did not really see any beads deposited here. And so for the aerosol chamber, the mice were continuously inhaling a 100 microliter diluted bead suspension. And so the main pattern observed here is that we do see beads being deposited into the lung lobe. Here you can mostly see them deposited into the alveoli. And as you can see, they're not really clustering together as we did in the previous slide. Here we're seeing them being like uniformly distributed throughout. So this is for the alveoli. And then here we do have an airway, and you can also see that the beads are being deposited. They're probably clustering maybe 1, 2 together, but overall they're not clustering as much as the large dose intranasal exposure. And here we do have another small airway and the alveoli showing the same pattern. And so to essentially quantify and understand our data, we incorporated the Python grid analysis method. And so for these two studies, I have the small dose and the aerosol chamber exposure studies here. For this image here, I have the overview image of the lung lobe with the grid overlay. And then here I have the black and white scheme. Then here I have the same scheme with the grid overlaid on top. and this is the purple numbered mask scheme with the numbered grid. And so this was all generated using Python. And essentially what we're trying to achieve here is for these two schemes specifically, we want to be able to observe these patterns and the distribution of the beads only. And the reason why I chose these two studies to present on this slide is because you do see fluorescence here and even in these pictures as well, but this is mostly from the tissue fluorescing. And so in this next slide, this is from the large dose intranasal study. Even though it's very, very hard to see, the fluorescence here comes primarily from these beads here that were clustered. And you can also see that here. And this is primarily what we want to use this grid analysis for. Furthermore, there's a lot of grids here, but the purpose of these grids is to essentially be able to compare the bead density of the beads that are in the lung lobes with one another and just compare and contrast that with different grids in the image. And so in conclusion, the aerosol chamber did show a uniform bead distribution in the lung lobes, whereas the intranasal bead exposures did have varying patterns. The grid analysis method does show potential in identifying these various patterns. And so for future directions, we are just looking to quantify this analysis using the Python grid analysis method. For these two future directions, we expose the mice to LPS, which is going to induce inflammatory cell responses. The reason for this was to provide more clinical relevance for this study. And so the only thing we have left to do is to interpret our image data. And we hope to do that once we have a quantitative analysis method to incorporate with the Python grids. And so lastly, we want to answer if the grid analysis method can compare differences in the bead patterns for the lobes of different mice, whether they're in the same study or in different studies. And so lastly, I would like to thank Dr. Lowe and Kazaya for their mentorship on this project, and of course for Ryan and Chi for their collaborations with this project, primarily with the aerosol chamber and for the Python analysis. And lastly, I will also like to thank the various programs I've been a part of during my time here, Marquis Star, Camp, and the Honors Program. Thank you so much for listening. I will now take questions.
Thank you, Martha.